So it's my pleasure to introduce, oh, hang on, got to get out of the way of that. Oh, Genevera Allen, who's, I think, the newest professor of statistics at Columbia. She started in July, and she's also an investigator in the Zuckerman Institute, which is the MBBI just up the street. Prior to coming here, Genevera was at Rice in the Department of Electrical and Computer Engineering and in statistics and computer science. Her research involves statistical machine learning tools to help people make reliable discoveries from large and complex data. She's known for her methods and theory work in the area of unsupervised learning, interpretable machine learning, data integration, graphical models, and high-dimensional statistics. And as you can tell by her affiliation, she's seriously interested in solving real biological or scientific problems, focusing mainly on computational biology and neuroscience. Genevera has won several awards, including an NSF Career Award and several things at Rice. And most intriguingly, I'm not sure I should say you were named the Forbes 30 Under 30 Science and Healthcare list, but I won't say when, because that's inelegant. Anyway, she's also a fellow of the ASA and of the Institute of Mathematical Statistics. So it's a great pleasure to have you here, Genevera, and the floor is yours. Just need to give you the gadget.
Okay, thanks, Simon, and thanks everybody for coming and being here. First of all, mic check. Whoops, let me get this set up right here. Can everybody hear me on Zoom? Okay, if the mic's here. Can folks hear me in the room and on Zoom? Hopefully, we're good. Excellent. Okay, so I'm excited to be here and excited to talk to folks in computational biology. So a little bit of background. I am a statistician who works in statistical machine learning, and I develop tools to help people make data-driven discovery. So what I mean by that is I develop tools that are hypothesis generating instead of hypothesis testing. This could be unsupervised learning. I've done a lot of work in graphical models, clustering, dimension reduction, etc. Also in supervised learning could be sparse models, feature importance, and things like that. And I'm very interested in data integration. I've done a lot in multi-omics in the past and so forth. But today I'm actually talking about a more recent line of research that I hope you will find interesting. And also, oh, and I should mention to any computational biologist in the audience, I'm very open to collaborations. I'm somewhat new to Columbia and love collaborating. So if you see or hear anything that might be interesting, please do reach out to me. And please ask questions throughout also. I love questions. Okay, so today I'm talking about more recent line of research on interpretable machine learning. And hopefully you will find this relevant for some of your work. So there's a big question that goes around. We've got these huge AI systems that are making critical decisions in many facets of our life and in healthcare. Of course, these AI systems are being used throughout science. We've had some people win some Nobel Prizes recently using AI systems in the sciences. But the question is can we trust these machine learning systems? And if you read the trust in machine learning literature, everybody says, well, make it interpretable and explainable to humans, and that promotes trust. And I say, okay, great, but can we actually trust those interpretations? Or are those interpretations reliable? And that's what we're going to be talking about today. So again, there's a large literature on trust in machine learning, but it's really focusing about predictions, not about interpretations. And so I talk about the 3 R's as prerequisites for trust in ML, AI, and largely computational systems in the science. Reproducibility, can we get the same results on the same data set? Replicability, can we repeat the study on a new data set? And reliability, can we obtain similar results if we have new data? And reliability is our focus here today. And in the statistics community, there's been a great recent work called conformal predictions or conformal inference. And these are basically uncertainty quantification and confidence intervals for predictions in machine learning. And this has grabbed a lot of attention in the past 5 years and is a very hot topic in statistics and ML. But the question is, what about ML interpretations? Again, all of this is for predictions. What about interpretations? And there's a few that have investigated the reliability of specific ML interpretability techniques, and they all suggest, hey, maybe these aren't as reliable as we thought they were, but there's really no large systematic studies and no way to actually look at all of these. So briefly, I just want to briefly kind of lay the groundwork for what do we mean by interpretable machine learning, and then hopefully you can see how this is relevant to your problems. So I define, first of all, there's a lot of debates in the field about how you exactly define these. These are my personal definitions that I've been working with. I define this as the set of techniques that generate human understandable insights into data, the learned model, which was fitted to the data, or the model output, the predictions. Okay, so importantly, the data, the model data interactions, and the output or predictions from the model. So this means we can interpret not just the predictions and the output, but also the data itself. And there's lots of different techniques that have been developed. There's a huge taxonomy of methods, intrinsic versus post hoc interpretations that are part of the model or interpretations that come after the model, model specific versus model agnostic. Model specific interpretations are inherent to one machine learning method. Model agnostic can be applied to any method. Global versus local, interpreting all observations in the model versus interpreting one observations. So again, there's a huge literature on this, growing by the thousands almost every year, on techniques for interpretability in ML. And getting back to the sciences, we can use these wide variety of techniques to make lots of different types of discoveries from data. And so while a huge amount, especially in the CS community, has focused on supervised interpretability, I would argue that all of unsupervised learning is really an interpretability. We're trying to interpret the data and the model data interactions. So we can find groups via clustering, patterns, dimension reduction, manifold learning, associations, graph networks, anomalies, and prototypes, maybe even motifs. Supervised learnings, we might find features that are important, feature selection, feature patterns, influential points, and so forth. So there's a lot of different types of scientific discoveries we can make using these methods. But a question is, are these types of scientific discoveries that we make using big, often complex black box machine learning methods, are they reliable? And so to motivate this kind of from a computational biology perspective, I want to highlight this paper here, this recent paper that is looking at, this is a single cell sequencing, and they also combine this with time-lapse microscopy to actually understand the cell cycle and transitions between cell states, okay? And this is a pictorial representation of what they put forth as the definitive. Here is the cell cycle in humans. And how did they get there? They went from kind of cells by features, and they applied kind of three main classes of ML methods. First, they did feature selection, feature importance. They found features that were related in the time-lapse data. They had some labels for certain cell states, and they found which features and which genes were indicative of the different cell states. So they did a feature selection step. Then they did manifold learning. That's how we get this like pretty picture here of the cell cycle structure using FATE. You can also use UMAP, lots of other manifold learning techniques. And actually on the cell cycle structure, then they did some additional clustering in an unsupervised manner to find kind of transitions and substates within the kind of cell cycle framework, okay? And the question is, they did a lot of machine learning techniques. Many of the feature selection, feature importance was done with random forest and neural networks. The manifold learning was done with FATE. The clustering was done with a graph-based spectral clustering method. Many people would consider such techniques black boxes, right? Like not necessarily, it's unclear whether these are reliable. And in fact, a follow-up paper from this group took the same data set and said, what if we actually looked at the feature importances and selected features via different approaches than what was done in the original paper? And look at all of these different manifold visualizations. All of these are using 30 features, but each one used a different technique to find the 30 features before doing manifold dimension reduction. And look at this, the interpretability would have dramatically changed. The science that was found and discovered would have been dramatically changed. So these results in the science we're getting are going to differ based upon these different ML interpretations. So I argue it's critical that we've got reliable machine learning interpretations to do rigorous and reproducible science where we can really understand that our methods are giving us kind of correct, reliable scientific discoveries to go upon. Okay? So given this motivation, our objectives are as follows. So I'm actually talking about kind of 3 separate papers within this talk, so please bear with me. The first one is a reliability study on are ML interpretations reliable? Let's first test this and see whether they're reliable or not. And then after this, okay, so if they're not reliable, what can we do? We're going to say, how can we validate ML interpretations in a data-driven way, something that's practical that you can take with you and use on your data? And also, finally, and this is the one I'm most excited about, how can we conduct statistical inference to quantify uncertainty in those machine learning interpretations we get? And so far, my group has worked on feature importance inference, and I'm going to briefly mention some of the other approaches we're currently working on. Okay, let's get started. Reliability. Okay, so we designed a reliability study because there's really been no systematic study of the reliability of machine learning interpretations. Again, there's a large literature on this for predictions, but not interpretations. So we wanted to set out to do this. And we asked 3 questions. The first one is, are ML interpretations internally consistent? Meaning, if we just had a different training set with the interpretations of the same method on just a different training set from the same population, yield similar results. So if an interpretation is reliable, the answer should be yes. They should be internally consistent. Question 2 is, are the interpretations from different methods similar? And specifically, if we apply 2 different techniques to understand feature importance, and the 2 techniques are supposedly finding the same thing, then hopefully their interpretation should be pretty darn similar on the same data set, one would hope. And finally, many have suggested that if we have a good supervised, and this third question is only applicable to supervised learning, if we have a method with much higher predictive accuracy, then we can certainly trust those interpretations. If the predictions are good, the interpretation should also be reliable, and so we want to test this. So there's a lot of different interpretable machine learning methods. So in this study, we needed to limit the scope, and we specifically consider tabular data and global interpretations only, in part because this is used a lot in science for scientific discovery. And we're going to consider 3 different tasks. We're going to look at feature importance in both regression and classification, clustering, and dimension reduction on the unsupervised side. And again, we have a lot of different ways, excuse me, to do this. What we did is we took, I think, like 38 different benchmark data sets in the commonly used in the machine learning literature, and we perturbed the data in lots of different random ways, random data perturbations. We applied over 50 IML techniques, and then we have lots of different ways of measuring the consistency and the reliability of these techniques. And just to show you an example kind of the study design and how we set this up, we took our data set, one of the benchmark data sets, and we randomly would split it into a training and test set multiple times. So we're just resampling which is the like 70% training and 30% test. And we do this repeatedly. And for each time, we apply the interpretation technique and we get the predictions. And then for question 1, we see basically on different random training set splits, are the interpretations similar? For question 2 is on the same split of the data, are 2 different techniques that should be giving me the same interpretations, do they give similar? And here we look at the kind of correlation between the prediction of and the consistency of the predictions and the consistency of the interpretations, okay? And I'm only going to present a subset of the results specifically on feature importance ranking using the top K rank overlap and clustering using the adjusted RAND index as my consistency metrics. Okay, so because I can't put all the results on slides, that would be crazy. Okay, So here's a summary figure, and let's delve into this because this is a big figure for feature importance and classification. Okay, so let's delve in. Question 1 is internal consistency. So are these methods internally consistent on like 2 different random splits? So what I'm showing you right here are different, we did 100 different random training and test set splits. And I'm showing you results of a subset of our methods on the x-axis, and on the y-axis are a subset of the benchmarks. These benchmarks are ordered here from large P to N ratio. P is the number of features, so high dimensional, down to large N compared to P. Up here, PanCan, DNAs, TCGA are all genomics based data sets. So you can kind of see what's going on here. And here I'm showing you the interpretation consistency. Oh, these little skulls means that method did not finish in 8 hours. We had to cut them off because, like, oh my goodness, my student, Luchin Gan, my former student who worked on this project, she like killed my servers every other weekend, running all of these like empirical studies. So we had to cut off some methods. That's what that means. And what we see here is higher values, like a 1 means they are always exactly the same and consistent. The top K features are the same, and 0 means they're very different across samples. And what we see here, sorry, I'm turning up my brightness so I can see the results too. So what we see here is that for some data sets, the methods are largely very inconsistent, but there's huge discrepancies. Some are better than others at certain points. Look at this data set, this author data set. There are some methods that are pretty consistent and some that are terribly inconsistent. But when we look at these same methods all give basically the same predictions and the same prediction accuracy. So over here, our prediction accuracy for all of these techniques are pretty good, yet the interpretations are not internally consistent really at all. We'd want to see this as like a dark teal color, and there's an awful lot of light colors here. And you might say, well, is there one method that is more internally consistent than every other? And to look at this, we just ranked the methods by how well they're doing across a range of benchmark data sets. And we see here, this is called a bump plot. And a bump plot, if there was a rhyme or reason, we should see something that like sorts out and doesn't look like spaghetti. But this looks like spaghetti, which means basically there's no rhyme or reason. We can't say there's one method that's definitively better than others in terms of feature importance. So this is kind of a negative result. And next we say, well, surely between methods, the results applied to the same training set should be consistent when they were applied to the same training set. And I really want you to zoom in here on, these are decision trees, XGBoost, random forest, and permutation for random forest. These are all tree-based methods using like MDI-based metrics for feature importance. Surely these are basically consistent. And look at these, all of these numbers are like 0.3, 0.4. They're basically giving vastly different interpretations and features, even though when we look across the consistency of the predictions, all the predictions are essentially the same. Every method is doing very well, yet look at the colors over here. Every method is giving different features that are important. Ouchers. So this should hopefully give you pause. And finally, we look at prediction versus consistency. And what we're plotting here is the consistency of the ML interpretations versus the accuracy of the predictions. And what we're hoping to see are positively sloped lines. Here we're plotting each point is a different data set, and we're plotting the different methods applied to the same data. And here we're doing the same data, each one is a different method, and each point is that method applied to the lots of different benchmark data sets. And basically, no matter which way you look at this, we're seeing a lot of flat lines, right? So the ML literature, which says that, hey, things that are more predictively accurate should be more reliable interpretations, we see no evidence of that in our study here. Okay. I'm going to pause here. Questions? Yes. Yeah.
Information on relevant models. what's actually in the data, like, there should be a good sample not to compare that and training model that way that is. What does that happen?
Oh, that's a great question. And in fact, and that's what I've been doing in my research, is taking an ensemble of many trained subsamples. And that turns out to work a lot better. But these methods don't intrinsically do that. And these types of interpretations don't intrinsically do this. Right? Yes.
I'm not sure.
Yeah.
Think something basic across that.
Yeah.
Do that by design.
Ah, okay, we're going to get to this. So cross-validation, can't cross-validation solve this problem? Well, think about it. Cross-validation decides based typically on, in terms of like the prediction error, some kind of log likelihood. And that's basically cross-validating based on how well the predictions are doing, not how well the interpretations are doing. And as we see here, the predictions are good. They're consistent. The predictions are good. So cross-validation is not caring about the interpretations. And it turns out we've certainly done empirical studies of this. Cross-validation gets you the wrong answer in terms of the interpretations. It does not help you with the reliability of interpretations. Yeah. Oh, yes. Yeah. Yeah, so the consistency index right here for feature importance is just the top K overlap, rank biased overlap. And what that is basically comparing the top one, the top two, the top three, and looking at the overlap between those sets all the way to the top K. The results I'm presenting now are the top 10 K. So this is a metric to compare sets of feature importances that's widely used in the literature. I should say we have, if you don't like our metric, we've got 10 other metrics and you can look up the results for. And if you really hate all 10 of our metrics, you can absolutely code up your own. I'll show you how to test your own out. Okay, so this is prediction. So let's actually go to clustering really quick. So here's a summary figure. I want to go somewhat quickly, but overall, some of the clustering methods were more consistent than others, but there's still some data sets, especially some of these high dimensional data sets that have a lot of kind of inconsistent clustering results. When we look at a bump plot, again, we don't see one clustering method as coming out on top as more reliable than everybody else. They're kind of still all over the place, which isn't the best. And in terms of the consistency between interpretations, we would hope that here the consistency means if you apply k-means and you apply spectral clustering, how much do those clusters overlap? And here the metric is the adjusted RAND index, which is a commonly used metric to see the overlap between clusters. And we see k-means and k-means plus plus. Okay, they're the same algorithm with different initializations. They're pretty consistent, but like k-means versus like spectral, they're not very consistent at all. So this is maybe a little bit of a caution also that we're not really getting consistent clustering results. So I should mention all of this raw data you can explore. It is available on a dashboard. This dashboard is a working dashboard, so please bear with us. You can fully explore all of our empirical results, both summary figures and raw data. You can upload your own data set to see if all of these techniques can be reliable on your data set. You can actually input your own IML method. It needs to be in the category of feature importance, dimension reduction, or clustering, but you can upload your own method, code for your method, and it will run. You can also upload your own code for your own consistency metrics if you don't like any of ours to try these out and see whether methods are reliable on your data sets and your methods. Okay. Okay, so just to summarize these results, our three questions were, are they internally consistent and reliable? And the answer is generally no. Are the interpretations from different methods that are supposed to be similar, are they actually similar? And the answer is definitely no, they're not. And finally, in supervised learning, do methods that have higher predictive accuracy have more reliable interpretations? And the answer was, again, a big fat no. So Youters, by the way, this is not exactly what we were expecting, negative results when we started this study. So I think to summarize, gives a lot of pause, right? We need to interpret with caution when we apply these types of techniques to very large data sets. But of course, I'm a statistician, so I don't want to stop there. And the question is, what can we do? And I'm going to address this from both a practical perspective and also from kind of a research perspective in terms of developing the next generation of techniques that can address these problems. So let's first address this from a practical perspective, because when I work with scientists, I always say like, hey, I'm going to go off and develop some big fancy like machine learning techniques. And I mean, these approaches, man, I might spend the next 5 to 10 years developing that, but you're sitting there as a scientist going, well, what can I do in the meantime to make sure that the results I'm presenting are more reliable and my science is rigorous and reproducible? So I want to at least outline a couple of things that you can do now to improve the reliability of your results. And this is related to your question. How can we validate ML interpretations? And in general, it's much more challenging than prediction. We don't have prediction error. Even as I was mentioning, cross-validation is based on prediction error. We saw the predictions are great, but the interpretations might not be great on the same data. So that is a problem. So there's two approaches that we can apply now. Stability, and that's directly reporting the reliability results, just like we did in the study we designed of your ML interpretations. And this shows the variance of the ML interpretations. But we do also typically want interpretations that generalize to new data. If we find clusters of cell states and the cell cycle in one data set, we don't want that to hold for just that data set. We want that to hold for other data sets that are also studying the cell cycle. If it is true scientific, it's a true scientific finding, it should hold for future data. And so to do this, we need to assess how predictable those interpretations are on new data. And this captures the bias in ML interpretations. And so I would argue both are critically important for actually assessing and validating our interpretations in practice. So first, stability. So the idea of stability is very similar to how we set up our reliability study. You're going to take your original data, you're going to randomly perturb it many times, say 100 times, 500 times. You could do repeated training and test set splits like I presented. You're going to apply your interpretable machine learning technique to make a discovery D1 to DB. And then you're basically going to say, we're going to record the discoveries that are the most prevalent, that are the most stable across all the random perturbations. These are the interpretations that are the most reliable, okay? So this is a somewhat familiar concept. I like this. This is model agnostic. You can apply this to literally any machine learning method out there. And when we apply this in clustering, just to show you an example, this is often called consensus clustering. So in clustering, we might apply this. We might take a subsample, a repeated subsample of our data, apply a clustering technique, and we might record how often two samples were co-clustered together. And what you're visualizing is often called the consensus clustering matrix. And here we're plotting the samples by samples in the PanCan data. And dark green, this kind of forest green, represents a 1, and this light color, oh, it's very hard to see. I'm seeing in the projector. It's not as clear as, oh, sorry about that. That's a projector issue. So this light color here is a 0. And we want to see if there's very, very clear clusters, there should be kind of this block diagonal pattern. And we do see that here, but there's some off diagonal confusion here also. And to visualize this data, we also plotted this in principal component space. And we plotted each individual observation here. The size of the point is proportional to how reliable that observation is clustered. And what we see here is, well, there's one red guy that's a little bit unreliable, but it's really, there's confusion between the blue and the teal clusters. The yellows and the pinks and the reds are pretty darn well separated. And this gives us a lot of information about the reliability of our clustering results and might tell you scientifically, hey, the blue and the teal guys, maybe there's not a clear distinction between those two clustering techniques, okay? So maybe a little computationally intensive, but this is something you could easily apply now. This is an example on clustering, but you can apply this to any machine learning interpretation. The next is generalizability. And the question is, how can we reliably predict our interpretation? So imagine we've got a training and a test set, and we're going to exploit that kind of paradigm of machine learning. And we might generate an interpretation on the training set, generate an interpretation on the test set. But how do we map those two interpretations? If it's clustering, we've clustered the training set, we've clustered the test set, we got to map them between it. So on the training set, after we cluster, we're going to take those labels and we're going to assume those are the truth and build a multi-class classifier on the training set to build a predictive model. And we can use those predictions and apply it to the test set. So now on the test set, we've got our interpretation, our clusters, and we've got predictions from a multi-class classifier. If something is really generalizable, those two should be exactly the same. If it's not generalizable, they should be a bit different, and we can compare those predictions. Again, this is a very general framework. When this is applied to clustering, some people call this clustering prediction strength. And here, this is the same PanCan data set, and we're showing you the training set clusters here in principal component space. And on the test set, what we did is we basically took these labels as the ground truths. We built a random forest classifier to classify on the test set. We separately classified on the test set. And we're showing you here the large points are all the observations in which the test set clusters and the predictions from the training set disagree. And the adjusted rand index is 0.7, which is okay, but it's not great. And we really see, again, it's the blue and the teal clusters. We're seeing the same type of thing, that those blue and the teal guys are not very well separated. And there is some confusion about these results. Okay, I'm going to pause here. Any questions? So this is a kind of like, if you are concerned about the reliability of your machine learning interpretations and you want to do something about this now, these are two approaches that you can apply right now to test the reliability. But what I really want to get into, and what I'm excited about research-wise, is inference now. So the question is, how can we quantify the how can we use statistical inference to quantify the uncertainty in ML interpretations? And specifically, we don't want to do so in a way that's model specific, because in machine learning, we don't want to use a specific model. We want to apply this to all clustering techniques or all supervised methods and feature importance. We don't want to assume a specific population generating model, because again, we don't know what that is in machine learning. And also, we don't want to assume anything about the underlying data distribution. So how do we do this in a completely kind of model agnostic distribution-free way? So far, my research has focused on feature importance, and I'm going to present results and how we did this. And then currently, we're moving on to some other methods as well. So again, our goal is to do this. And this has been done, right? Doing kind of inference for ML interpretations. Like, let's use clustering as an example. If you assume a Gaussian mixture model and you apply a Gaussian mixture model method to do this, you can directly, when you assume a specific population model, a specific data generating mechanism, in this case, a mixture of Gaussians, and a specific method, you can easily get inference, right? And you can do this in Bayesian settings as well by assuming specific priors and so forth. But how do you do this when you're not assuming any model or any data generating mechanism? That's the trick. And that's what would make this like actually really important to do in practice and really useful. Okay, so there's been some related work. I mentioned this for predictions, right? Like in ML, everybody studied predictions really well. And this is conformal inference. And specifically, we could get distribution-free and model-agnostic confidence intervals for predictions. And there's been a lot of work on this. This is a very exciting area of research in statistics and machine learning. The question is, can we do something similar for feature importance, which is an interpretation, but not a prediction? So feature importance inference. First, I think we need to define what we mean by feature importance. So feature importance is typically defined as how a feature J affects the output or the predictions or your predictive model f of x. But when you dig deeper, most people, when they talk about feature importance, especially in the context of inference and in the statistics literature, They're talking about this on the right side. Is that the right side to you? Whatever, yeah, right side, cool. They're talking about population feature importance, and specifically, this is how does feature J affect F star? So if you fit a linear model, for example, using least squares, and you're testing coefficient beta hat J equals to 0, you are testing a population model. You assumed a linear model as your F star, and your statistical inference is for that population model F star. So this assumes a data generating model, but it's a property not of the specific ML method you applied, but really of the underlying model itself. ML feature importance, I argue, is really what we're after here. And this is a little bit different. It's how does feature J affect F hat instead of F star, where F hat is your machine learning model that you trained on your data set. So this is your random forest model. This is your deep learning model that you built. How does feature J affect your F hat? Okay. And this is a property of the model of your kind of F hat data interaction. So this is a little bit different. And when it comes to inference on these things, again, there's a lot, all of classical statistics, all of statistics is on this side. And more recently, there's actually been a very exciting line of work on inference in machine learning context using occlusion, which we're going to get to. And several of these great papers here, but they are all doing this population inference on F star. And actually, there's a great paper by Sean Peters that showed that to do this and have non-negligible statistical power, you actually have to either make strong assumptions about how close your F hat is to F star, otherwise you can't say anything about F star, makes sense, or you have to actually have knowledge of the data distribution of your data. And that this type of inference is impossible without one of those assumptions. But those are particularly what we're trying to avoid. So instead, there's a line of work called LOCO, or occlusion inference, leave one covariate out inference, on ML feature importance. And this is closer to what we want. And so this is kind of the framework we're going to work in. Okay, so LOCO, what is this doing? This is called leave one covariate out inference, leave one feature out inference. The idea is really simple. We are going to leave out feature J and refit our F hat, and we're going to see how our F hat and how our predictions change when we leave out feature J. If feature J was critical, our predictions and our prediction error should change a lot. If feature J was not critical, we shouldn't see a change at all, okay? So in this approach, they looked at these feature occlusion scores by doing data splitting and conformal inference. And specifically, they proposed this inference target. And so notationally, just bear with me, but this is looking at the error. This is any prediction error. Oh, I should use my pointer so people on Zoom could see. Okay, so this is the prediction error, and this mu is our prediction function. Okay, so any prediction function, and here this is fit using all of our data. This is a prediction on a new point X, given our training data, X boldface and Y boldface is our training data. And this is fit without feature J. So if you stare at this long enough, this delta star, if feature J is important and changes the prediction error, this delta star should be strongly positive. But if it's not changing the prediction error, it should be 0. So it's the difference in the prediction error with and without feature J. Yeah. This is a great question. I'll repeat this for folks on Zoom. Correlated features, what do we do about it, right? Like, this is a fantastic question. This is an ongoing debate in the field. Let me get back to this if I have time, hopefully, and get back and answer your question. Okay, cool. So their approach and how they did this is they split their data into a training set and a test set. In inference world, this is called a calibration set. They fit the predictive models, these mu's and minus mu's, to the training set. And then on the test set, they use the residual differences to construct the confidence intervals. Seems completely reasonable, seems nice. There's several advantages of their approach. It's truly model agnostic. They only studied it in proof theory about the regression case, but it's generally model agnostic, very assumption light, and it truly is distribution free. Makes no assumptions about the population model or the data generating mechanism. However, at first when I read this paper, I was super excited. I was like, this is really cool. However, there are some disadvantages. First, in the inference literature, we know that data splitting always loses statistical power. In the context of getting confidence intervals, this means wider confidence intervals. It's pretty computationally prohibitive, especially if P is, you know, tens of thousands, hundreds of thousands. You're leaving out each one and refitting. That could take a while, so it could be computationally a problem. But the third point is really the kicker for me. They split their data into a training set and a calibration set. Their inference is conditional on the split of their training data. Okay, let me repeat that again. Their inference is conditional. When we go back to this inference target right here, when they split their data, it's really conditional on X train, Y train, not their original full data set. So what does this actually mean for interpretation? This means if you split your data, we just looked at the kind of reliability of ML interpretations across random training set splits. If we've got two different training sets, we could get two completely different answers. Furthermore, if I go to scientists and I say, oh my goodness, I'm so excited, I got this confidence interval for you and this p-value for you, but it's conditional on this random split of the data, and you need to use this seed, 10071. And if you use a different seed, you're going to get a different p-value. How many people in this room think that's okay to do? An okay type of inference? Oh, hopefully nobody thinks that's okay. Okay, because I am disturbed by it, and I hope other people are equally disturbed. So this point 3, we said we just can't do data splitting. We can't produce an inference result that's conditional on a random split of the data, right? We need to actually use the full data to produce our inference. So our idea was, okay, so I've been doing another line of work called mini patch ensemble learning, which is super fun, by the way. What we do here is it's kind of inspired by random forest, and we're taking double subsampling of the rows or observations in our data set and the columns or features in our data set. So something like this, and when you permute the data, you get this little patch here of data. So we call these mini patches, not to be confused with mini batches in stochastic optimization, which take all of the observations. Here we get both double subsample of observations and features, okay? And we're going to do this a bunch of times and take a bunch of different random mini patches, okay? So why might we want to do this for inference? So the key thing here is to do inference appropriately. We need to leave out some observations for a calibration set, and we need to leave out some features. And look, there's big white chunks here. But we can't just actually do separate inference for every mini patch. Can we cleverly combine them in a way that allows us to do inference on our full data set instead of a random split? And this is kind of our idea here. So step 1, we're going to fit a random mini patch ensemble learning method. Oh, and you can fit any kind of learner that you want as your base model. This could be a decision tree, and you get kind of a random mini patch forest, something like that. You can fit a small neural network, and when you combine them, you get a larger kind of deep learning system. I mean, you can fit whatever you want. So fit a mini patch ensemble. And our inference approach requires no additional computation. Here's what we're going to do. We're going to say that we're going to average over all the mini patches that don't contain observation I. We're going to call this the Lou estimator or the Lou predictor. Leave one observation out, okay? And then we're going to get the Loco Lou estimator. And this is going to be leaving out one observation I and one feature J. And we're just going to sum over all mini patches that left out I and left out J. Okay, so we're not refitting anything. We're just kind of looking at the indices and all of the different mini patches and finding all the mini patches that didn't contain observation I and then jointly didn't contain observation I and feature J. And based on these, we're going to propose these feature occlusion scores, the prediction error of the Loco Lou estimator minus the prediction error of the Lou estimator. Okay. And then we can look at these over the entire training set. And we're going to then construct asymptotically normal confidence intervals for delta J. So importantly, all the computation happened in step 1. There's no additional computation we're running. We just are keeping track of the indices to get these estimators, to get these feature exclusion scores. Okay? So our inference target here, so the interpretation of what we're doing here is how important is feature J when you're deploying that specific F-hat model built on this specific training set. And in particular, we show that our mini-patch feature inclusion scores are indeed conditional on the entire data set, not a random split of your data via data splitting. And again, this is free computationally, no model refitting, and there's no data splitting involved. Okay, yes. Yeah, so the model, so let me go back to the picture of mini patches. So here is our mini patch. And what this is doing is we've got kind of, we typically do this over 1,000 mini patches, say, okay? So we're going to take this particular observation and we're going to look at all of the mini patches that don't have this observation. It's like a left out holdout set. It's like leave one out, cross-validation, for example, or the out of bag error in random forest. We're leaving that one observation out. And so we're not having to refit anything. We're just summing over all the mini patches that don't contain observation I, and that allows us to make a prediction on observation I and it be an unbiased estimate of the prediction. Yeah, so this inference approach, after all the computation is in step 1, this inference approach is just keeping track of the indices for all of the mini patch ensemble members. Yeah, I mean, okay, so there is a cost in doing all of these mini patches. I mean, we typically run 1,000. Sometimes if it's a big data set, 10,000 mini patches. There's a cost, but it's embarrassingly like parallelizable and distributed, right? So like think about, it's about the same cost as a random forest, right? Random forests are not doing something that dissimilar. So it's about the same computational complexity of a random forest, which I think is a pretty doable computational complexity in practice. Yeah. Okay. So, and then we just construct an asymptotically normal confidence interval for delta J here based upon these. Again, we're just getting the sample mean over all the observations. And again, you can really see this is conditional on the full training set. On all n observations of the full training set, we get an asymptotically normal confidence interval. But the question is, are these valid? Does this give you a valid confidence interval for delta star J? And this was a really big theoretical challenge. Let me tell you, the empirical stuff of this paper was done like 3.5, 4 years ago. I mean, like the theory turned out to be quite tricky. And it's because each of the delta hats for mini patches are dependent. In fact, 2 mini patches share often like n-2 samples. They're super dependent. And this breaks all existing central limit theorems. Like it just breaks them. It's too dependent. So there's a question of, what do we do here? How do we solve this problem? And so we obviously solve this, and that's why I'm giving a talk about it, and under certain assumptions. And we need 4 assumptions, but they're pretty darn mild. We need the error function to be Lipschitz, to satisfy a Lipschitz condition. This is either trivial or always satisfied, depending on which prediction error function we use. We need bounded mini patch prediction. So the difference between the predictions of any 2 mini patches can't be infinite, okay? So this is always satisfied for classification. And under regression, we need to make sure that we don't get infinite predictions. So it's basically a always satisfied condition under real functions. And mini patch size and number, we need the number of mini patches to be somewhat large and the mini patch size to be somewhat small, okay? And under these conditions, we proved asymptotic normality of our delta hat estimators. And we also proved that our confidence intervals have 1 minus alpha coverage for these confidence intervals. And so you might say, well, how on earth did we prove this? And there's something called, it's really important in the conformal inference literature called algorithmic stability. And you need basically algorithms to be stable in order to get some of these theoretical results and validity to hold in these situations. And the main part of the proof, and I should mention a former postdoc of mine, Lily Zhang, did the heavy lifting of this proof. The main thing that we proved is that by mini patching itself, the mini patching will stabilize any base model enough that algorithmic stability kicks in. And so we can leverage some of the other existing literature that is not satisfied by other methods. So basically, mini patching gives you more stable estimators, and that allows you to circumvent some of the issues. And that's how the proof worked. So I should mention, we did a lot of other things in this paper, including if you don't want your mini patch size to be small and you want them to be large, we've got an alternative set of assumptions that work for any mini patch size. We also show valid coverage after hyperparameter tuning. This is a really big deal if you can tune your hyperparameters and still get valid confidence intervals afterwards. And we showed inference, how this relates to inference for the population target, meaning inference for F star instead of F hat. And we also discussed this with correlated features, which I said if I had time, I would get back to. Okay, so briefly, by the way, we can use loco stuff for prediction intervals, for conformal prediction intervals, and I'm totally skipping that. Okay, so valid coverage, we did some simulations, we checked it, yay. Interval width, we're comparing our loco mini patch estimator in red to loco split method in pink and loco split with mini patches in purple. You see that the interval width is on the y-axis, and ours is much, much smaller, as we would expect, because we're not losing statistical power with data splitting. And just to show you a subset of the simulation studies and confidence intervals, this is a random forest under a linear simulation and a random forest under a nonlinear simulation. And our confidence intervals for feature 1, this is the SNR of feature 1. And this is a larger study, but we're showing some of the zeros here, are shown in red and loco split is in gray. And the main takeaway is our intervals are much, much smaller. And you can see that ours are above 0. So you would say any of these features that have a confidence interval bounded above 0, where the lower confidence limit is above 0, would be statistically significant or interesting. And loco split just does not have the power with much wider confidence intervals. You're not able to see those results. So correlated features. Whew, okay. I'm running low on time, but I really want to answer this, so I'm going to answer it. Okay. So correlated features. It's a complicated story. There's a huge debate in the literature. How should you do feature importance, and what does feature importance inference mean when you've got correlated features? And some argue that you should be looking at the unique contribution of feature J. So that is feature J given everybody else. So that's exactly what occlusion would give you. Others argue, no, if feature J is correlated with a bunch of other features, then those other features are probably also important also. And I think especially in computational biology and in neuroscience, we think those correlations are important and we want to pull them out. So I'm in the camp of, if features are strongly correlated, we want to pull strongly correlated features out. So we actually proved that mini patches actually take care of some of the issues with correlation itself. What we specifically proved was that in the mini patch loco estimator, if two features are strongly correlated, their delta, say feature J and K are strongly correlated, their delta JK is close together with the mini patch occlusion estimator, but not with the loco split estimator. And we proved that this is a property of mini patching itself. So we see this, let's look actually at this random forest setting. So here feature 1 and 2 are strongly correlated. And sure enough, loco split says feature 1 is important, but feature 2 is not, because they're super strongly correlated. So if we have feature 1, loco split says we don't need feature 2, whereas ours says we're going to pull both of them up because they're both strongly correlated and they're both important, and ours actually pulls them together. So I think this is a very strong result, although I presented this to some statisticians who were very angry at me on this slide and said, no, this is not the correct thing you want to do. Up for debate what you want to do. I argue in science this is a useful property. Okay, let me conclude with just a quick case study, a preview of a quick case study. This is a ROSMAP study, which is a longitudinal study on aging and Alzheimer's disease, where they had collected a ton of different types of data sets. We're specifically looking at the post-mortem genomics, so bulk RNA sequencing on post-mortem brains. And specifically, we're saying from postmortem gene expression, can we predict a cognitive score on testing that was 6 months proximal to the person passing away? Okay, so it's kind of a weird thing, and you would think you're not able to predict this well at all. We actually fit a random forest. We tried a bunch of different models. Random forest actually gave pretty decent predictive accuracy, believe it or not. We did aggressive gene filtering, and this was mainly for the comparison methods that we're showing, which were very computationally expensive. We didn't include all the genes, although we did in our study. So our method found, you might say, oh my goodness, that's crazy. We only found 3 genes that are significant, that are bounded above 0. Okay, so 3 particular genes. This sounds like not much, but I would argue in the computational biology, if you find 1,000 significant genes, you probably have 999 that are false positives. Sorry, I said that, but that's okay. But what we did to validate this is, actually, let me go to this slide because it's cleaner first and then explain it, then go back. So it's hard to tell when we apply several different methods that find different genes and different features, which ones are actually meaningful. So we took each feature from feature 1 to feature 50, and we ordered them according to either the lower confidence bounds or their p-values for those genes. And then we fit them one at a time into a random forest model. So we fit a random forest with 1 gene, with 2 genes, with 3 genes, and so forth. And what we should see is the test mean squared error should dip if we found the right set of genes, and then should hopefully go up. And sure enough, for loco-split, by the way, loco-split didn't identify any significant genes. It's really underpowered. You can't identify anything. Our method identified 3 significant genes, and you see the test mean squared error. This is on a separate test set that we set aside, is way, way smaller for these 3 genes, and then goes back up again, lending credence to maybe we actually found 3 really good genes that are super predictive on a separate test set. And then we compared to some of these other methods, the CPI, Floodgate, and GCM, and VIM, which are all methods for population ML inference in the literature applied to random forest. The VIM didn't find anything significant, and we think all of these are miscalibrated. One of them found 28 significant genes and so forth. But look at these. They're all finding significant genes as they're going back up again, and none of them dipped the same way ours do. They did not find the same top 3 genes that our method did. So we do think somehow this is finding something interesting. Furthermore, when we examine these genes, and this is an ongoing case study, so we haven't really finished this up yet, but the first 2 genes we found have been implicated as upregulated in Alzheimer's disease. The 3rd gene is perhaps more interesting because this is a long non-coding RNA, and there's no known links at all for this particular link RNA to Alzheimer's disease, but there's a growing literature, a very recent literature, linking these regions to other neurodegenerative diseases, including Parkinson's and other forms of dementia. So this is potentially an interesting one to study because it is very, very predictive in our study here. Yeah. That's right. It's post-mortem brain tissue, but it's cognition before death. Oh, we just left out a separate test set. Yeah. So we had a separate test set to train on. We trained on different data set and applied this to a different test set. That's awesome. We just left out like a third of the data, right? So that we did have measures and we knew how well we were doing. Yeah. Yeah, it is based on their clinical information before they passed away. Yeah. Yeah, it's a little bit of a weird data set. And if the fact that we can predict anything at all and find anything significant is pretty interesting, right? Like, because most people didn't think this was doable, right, on a data set like this. Okay, let me quickly summarize these. I'm a little bit over time. Okay, so hopefully I convinced you. ML interpretations, maybe not quite as reliable as we would hope. We really need to validate them in practice. So I always say to scientists, what can you do now to help validate your ML interpretations? You can use the stability principle and generalizability, and you can apply those now. So my current area of research is how do we develop confidence intervals for these ML interpretations? I presented some results on feature importance. What my group is currently working on now is inference techniques for clustering and dimension reduction. I'm very interested in getting inference for clustering techniques. Kind of, are these points well clustered? Are there features that are different in these clusters? This is very useful in single cell sequencing, for example, or spatial transcriptomics. Are there separate clusters? How well are they separated? What's the uncertainty there? And I absolutely think there's a ton of future statistical work and scientific work to do in this area. I want to thank some folks in my group. Lily Zhang is a former postdoc. She's now at UIUC. She did a lot of the heavy lifting on the theory for the inference. Luchen Gan is a former PhD student of mine who did the work on reliability and also the empirical work on the inference study. And Tarek is currently a postdoc in my group who is currently working on some practical validation strategies for machine learning methods and some references. And thank you very much for your attention.
Any questions? And everybody, Genevera, that was fantastic.
Cool.
Especially for those who have just been clustering.
Yeah, so sequential data like time series type stuff. So in sequential data, we have not applied mini patching yet there, but I think you could do this by basically taking short time chunks and then shuffling that way. So basically, you've got kind of windows of kind of fixed length that you keep the temporal structure and the sequential structure, and then you randomly sample all these little kind of like patches. So you just keep the kind of temporal structure. This is similar. There's a large literature on bootstrapping for time series, and it's a similar technique for bootstrapping in time series. And
There are things that are used at all features have but there is no thing uniformly for all. Would you be like another way of asking question? Would it be possible to assign different
oh, sure. Yeah. Okay, great. Yeah, absolutely. So in this talk and the inference method so far, we did random mini patch sampling, but you can actually do weighted mini patch sampling. And I think the theory would change a little bit. We need to update some of the theory for that. I think you can get a normality result, but it would change based upon the weights used. Another active area of research is we're doing adaptive mini patch learning. So what if you want to adaptively find the best observations and features on which to learn? I've done some adaptive mini patch learning for things like feature selection, feature importance, feature ranking, clustering, etc. And in all of those settings, it's really cool. The adaptive stuff works really well. We're working out how to apply inference techniques when we've adaptively learned the mini patches. It's a little bit trickier, but I do think it could be doable with some inventive theory and some grad students that want to work on the theory with me. Okay, yeah.
In the first part, when you're starting about is it possible say, oh, one subsample from one hospital.
Yeah.
No.
Yeah, so I definitely think that happens in real data sets a lot. So the empirical study we did was on benchmark examples used commonly in machine learning. And these are pretty clean pretty nice benign benchmarks, right? So most of those batch effects have been really carefully removed. So the fact that we're seeing these in pretty benign benchmark data sets is, you know, we would expect in real data, we'd see even more, and batch effects would affect that even further. So yeah, these were very clean data sets, well pre-processed, well studied in machine learning. Yeah. Oh, so
Yeah, probably things like time.
yeah, yeah,
Or, for example, the one that you write on.
gender. Yeah. That is, that's a great question. So I have not worked too much on kind of the causal type of inference and treatment effects and instrumental variables. I absolutely think these techniques could be applied and extended in that setting. I have not done so yet. It's not an area of expertise of mine, but would love to. It is an area of expertise if someone else is and they want to collaborate, would love to do so. I do think that these techniques could be extended in those settings.
Thank you very much indeed. That was great.
Thank you.